Very good morning to all of you. Welcome to the SERB Scientific Social Responsibility Research Facility Training Workshop on Immunofluorescence Microscopy of Amity Institute of Physiology and Allied Sciences. At the very beginning, I would like to call on dais our Dean of Health and Allied Sciences, Professor B.C. Das, sir. Next, I would like to call on stage Professor Dr. Jasovanta Sethi, sir, Director, Amity Institute of Physiotherapy. Next, I would like to call Professor Dr. Tanvi Navid, sir, the head of Amity Institute of Pharmacy. Our next dignitary on the dais is Professor Subhrajit Biswas, sir. He is the Deputy Director of Amity Institute of Molecular Medicine and Stem Cell Research, and also the Faculty Coordinator for Amity Institute of Physiology and Allied Sciences. Welcome, sir. We have another dignitary on the dais, Professor B.N. Malik, who is the Director of Amity Institute of Neuroscience and Neuropsychology, but he will probably join us a little bit later. So with this, let me start the program. At the onset, I would request Professor Subhrajit Biswas, sir, to kindly welcome our Dean, Professor B.C. Das, sir, with a flower bouquet. Okay. Next, I would like to request my faculty colleague, Mrs. Lakshmi Ramesh, madam, to kindly give a flower bouquet to Professor Dr. Tanvi Navid, sir. Next, I would like to request my faculty colleague, Dr. Sandhya Singh, madam, to kindly present the bouquet to Professor Jasovanta Sethi, sir. And I would like to request Professor Jasovanta Sethi, sir, if you can kindly present the bouquet to Professor Subhrajit Biswas, sir, if it is possible. Okay. Okay. So welcome again to all of you. I am Dr. Sudipto Saha, the Assistant Professor 3 of Amity Institute of Physiology and Allied Sciences. This particular workshop, the title, as you can see, it is SERB, that is DST SERB, DST Science and Engineering Research Board, Scientific Social Responsibility Research Facility Training Workshop. So this is a component when a project is received from SERB, they will provide a fund for doing such a workshop so that the faculties, the PhD scholars, and the students, they can understand that what are the different facilities that are available in that particular lab, how they can be used, and hands-on little bit of practice. But unfortunately, due to the COVID-19 pandemic, we could not do the hands-on. But now, as we are gathering now together, the COVID pandemic has gone down a little bit. So we have tried to do a workshop like this, which is in hybrid mode. So some are in offline, and many people are online on the Zoom platform. So we have selected the immunofluorescence microscopy technique to be displayed here. So we will be having certain talks, and then we'll be having some demonstration of the immunofluorescence microscopes and its softwares. So by this way, the faculties and the students will get exposure to this technique, and the scientific talks will also be, I hope it will be much enriching to the all the participants. So with this, I would request our Dean, Professor B.C. Das, sir, to kindly address the House.
Well, very good morning to one and all. All the students, participants, all the speakers, and also chairpersons on the dais who would be chairing different sessions. And I welcome all the people to this DST or SERB Scientific Social Responsibility Research Facility. This is a facility or this is a workshop. Generally, anybody got the SERB projects, DST SERB projects that is inbuilt in that project. First of all, I thank the DST to bring this point in because we always, particularly myself and each and every meeting, particularly related to the students or research scholars in the orientation and everywhere, and in personal level discussion, I always say that whatever you do research or study or anything, it should have some social context. Otherwise, we are being a social individual or a social person, so we cannot live without society. So your research or contribution, learning should help the society to grow. So if you are doing research, some research that should have the implications or usefulness to the society. May not be immediately. What you do research, it is not immediately that it will be translated to the bench to bedside, not. But in the long run, if you have this thinking in the back of the mind, so you can do a lot. So that keeping in mind that, so that you can do a applied research or a translation research, what we call. But you can have a big paper, big publications in Nature, Lancet or anywhere. But if it is for 100 years, it is not come to an use, then it is not a good research, I would say. You may have your career right, but that has no implication to the society. So we cannot do any research or any contribution in the vacuum. That's why DST should be thanked for this type of program. They have incorporated all types of SERB funded projects. You have to do 1 day or 2 day workshop to make your society around to the students, faculty and others, because this is a hybrid mode. Anybody can attend and listen to the talks. And I hope they will also put it in the YouTube and all that can be done. So this is today's workshop is before I go to this workshop or a little bit telling about this, first of all, with the in behalf of our Honorable Founder President, Dr. Ashok K. Chauhan, and our dynamic Chancellor, Dr. Atul Chauhan, and our very active Vice Chancellor, Professor Dr. Balvinder Shukla, and and on my behalf and on behalf of the Amity University, all the faculty, and also from the Health and Allied Sciences, because this program is arranged by the Amity Institute of Physiology and Allied Sciences, IPAS, because he got the first time a project from DST, Dr. Sudipta Saha. So that is why this workshop he has gone. I think more than a year the project has come. Yeah. So now he has organized this workshop. So I once again welcome all these people and all the participants and the speakers. He has got 4 speakers in this today's session up to lunch. So it's very compact and very nicely organized and nicely organized sessions. So But what is this? Today's workshop is on immunofluorescence microscopy. I think everybody knows it because it's a very basic and standard techniques. If you don't have anything, at least you have a immunofluorescence or immunological studies. But what does it mean, immunofluorescence microscopy? Because you do some microscopy with the help of what you want to detect, it is fluorescent level. So you either you level the samples with the fluorescent dyes. There are n number of fluorescent dyes, starting from the simple original. It can be of the protein staining, it can be of DNA staining. So what is your mostly it is looking for a protein that it is mainly the detecting or utilizing target antigen because it's immune molecules you detect. That's why immunofluorescence, that you fluoresce the immunomolecules or immune molecules which can be of protein, mostly protein and antigen, using fluorescence leveled specific antibodies. So you level one of the, that is antibody antigen reactions, and then you can detect where it is the, so that can be done in vitro as well as in vivo. On the slides, as you see the fluorescence microscope here, fluorescence microscope is essential with lot of, from the simple fluorescent microscope to confocal microscope, where you have the laser and the PACS machine of fluorescence activated cell sorter, where variety of filters and the lasers can be used according to your choice. Mostly it is the green fluorescence we see, which is derived from the jellyfish, which is autofluorescence. Many of the animals autofluorescence. You know the what is called what is that? हिंदी में नहीं जोनाकी Firefly. Okay. No, Hindi में कोई special name है. Jugno. So you see that is autofluorescence. Everybody must have, those who are from village. At least I have done. In Bengal it is too much because I was in village. So you will see in the evening, as soon as the darkness comes, you will see a lot of jugnu. And in the earlier, in the childhood, it's really a fun to catch hold of it, and then it's difficult to catch, but you catch it and then put it in the his head down on the gobar or mitti, then the soft soil or gobar, you put it, then it will go on, it will not go, it will always blip blip. So jellyfish is originally from which rather GFP has come into the green fluorescence proteins. So GFP is very big way used in all types of localizations in fluorescent microscope and confocal and everywhere. GFP is very much used in that you, and in the beginning, in the very early days, you used to see, I am basically student of genetics and cytogenetics from Banaras University. We used to stain the chromosome banding, or you know that was now banned to test the sex, the Y chromatin bodies and Y body or a bar body. The bar body you can stain by GIMSA and you can see, and there is specific training. But to see the Y body, that's very important, because in North particularly it was very common that they want to have a male baby. So you have to be sure, not that absence of bar body, but in amniotic fluid slide, but it is important to see the Y body is also there, Y chromatin is there. That is stained by the quinacridone or any other fluorescence, which is a DNA bound. So particularly those who are repeat sequences or heterochromatin. Chromatin are two types, euchromatin, heterochromatin. Heterochromatin is each of 80 reach chromatin. Some place they are GC reach also, but 80 reach or repetitive sequences take the stain like fluorochrome or fluoresceins or any names, rhodamine, and many variety terms if you want to stain red. So variety of, there is n number of the fluorochrome or the fluorophore are used for the like hex 33258 and 443, 334. So there are many types of fluorescence dyes are available. The main importance is to locate you and to detect your gene of interest or your protein of interest, your antigen of interest, and that is possible through this simple procedure. If you have a fluorescent microscope, there is a filter change also. You can have the different color and you can have contrast. But the problem comes to three things: that quenching. So if you stain, some of the stain quench very quickly before you take the photographs. And to enhance that, so there are several those who will be speaking in this specific subject will come to know this. There are not that it is easy, but then there are pros and cons, and the precaution has to be taken that if it is quenched, you cannot do. So what I was telling, that the Y chromatin also very difficult, but that is why important that you should be very experienced. Just now that it looks very easy, and when you go to do it, you will face several fluorescence. Which one is right? It can be staining other material also. So it is only experienced eye can do that, and it has happened havoc at the time of what I was explaining, the Y chromatin and bar body study. And it has been, I remember my friend, I don't want to take his name, because they used to connect me for sometimes, and he actually lost the job in a private company. His job was in a Dabur company. He was supposed to give the diagnosis of the, which is not officially approved, but that was 20 years or 30 years back. So he was supposed to give the diagnosis, and he came from abroad, but then he was diagnosing the Y chromatin, that whether the baby is male or female. And he diagnosed in such a way, he said that it is there are two cases. One is that it is a female baby, and they aborted and it was found to be a male. In the another case, they said it is a male baby by seeing the Y chromatin body, but it was not a specific staining or specific. You can, when you do, those who have experienced fluorescent microscopy, if you stain with the fluorescent eye, there will be a lot of staining, but experienced eye can make it that which is right and which is wrong. So many things can take the stains. So that is why it is very important that you have to be experienced with that. And he declared that it is a male baby, and ultimately it was a female one. So that was and that led to a lot of problems. So this can be done with any type of materials on the slides, and also the histology or histological slides, which can be stained to locate that antibody is leveled, and you can use that. So and live cells also you can do in the confocal conditions. So in the fluorescence and the facts. So there are several, I don't go into all these details. So these things are very basic things, and one should know it. And it is not that, but it is a very basic and the standard technique. Every basic laboratory facility, and I am thankful to Dr. Sudipta that he could create this facility because of this project. He was the only person in that IPAS who has got the project, but I expect there are new faculty joining, and they will be able to, they have been trying, but as soon as you get a project, you can imagine he will show I think a video presentation, his lab. He has only one project, and I had gone out of the way to support and convince Vice Chancellor that he is lacking the tissue culture facility. There are 2-3 things: the incubator and the inverted microscope is essential. And I could convince us, he has agreed, and I think it is 8 lakhs or 9 lakhs. 8 lakhs, it must have gone now order. I am also chairperson of the funding, the purchase, so I have also okayed it. So it has gone for order. It is not that the university does not support, but if you come up to their expectation, they will support. So that's why you will see from the video, he is only one project. 45 lakhs, 40 lakhs or 50 lakhs?
Just 30 lakhs.
30 lakhs. But he could intelligent, I will highly appreciate him that he put very basic things, small equipment, several equipment, so that he can build up a lab. And that is why now the people can work there, students can work there. Like that, if the 2-3 persons faculty brings one project, then the lab will start and lab will grow. It is not that you have to, that is what the credibility here in the MIT, that you have to prove your worth. And that is what we say during interview and all, that you should be capable of bringing the project. That is your proving yourself that you can have your own baby. So this is the means scientific baby, that you have your projects and get the grant approved. Applying, no replying, it's no fun. You can apply hundreds of projects, but if doesn't get success, then it is no fun. It means there is some problem with you, that you are not getting your project funded. So if you have a fund, you can play around with your hand, so that you can appoint PhD students, you can create your facility, you can get going. So you can have your mental picture that what you want to do research, you can start your own thinking, own project, and that is how you grow in the scientific field. So this is out of the track a little bit, but I said that it is important because this is the workshop being held because of his project. Otherwise, you would have not had the opportunity to have so many speakers to invite and to students or participants and all. So I will not take this microscopy, of course, very powerful technique, and it is simple, but you have to be experienced, but very quickly you can get experience. But it's certainly a very powerful technique, and it is starting from, it is a basic, from there to higher up to confocal to facts and many things. So once again welcome you to this workshop, and I thank Dr. Sudipta, and also this, though I am dean, there are every the health and allied sciences, there are 8 departments and 8 institutes, which are 8 coordinators are there, or 5-6 coordinators are there, which is our honorable founder president has decided. So this department is, or institute is coordinated by Professor Subhajit Visas. He is my colleague in AMSER. So they are all from AMSER, but just to I am busy for many other things of the university. So it has been that they can immediate need, they can talk, they can come across to each department or institute knows their coordinator. You can approach them, and when it is not solved, then you can come to me, and then we can solve together. So, but thanks to Sudipta, that you could get the project, and he has shown the path, and you could establish a good lab, and there are students working, and that's very good. And I think I thank all the health and allied sciences faculty, and all the members of the IPAS, those who have been helping and to make this workshop successful. Thank you very much. Thank you.
Thank you, sir. Thank you so much for inspiring us and enlightening us. So before going into our sessions, we have a very small video of the lab, because this particular workshop is, as sir has just now said, is because of this particular project. And through this project, we have developed a lab. So we have a small 2 minutes video of the lab to show that how the setup has been made. ये वाला नहीं, ये वाला नहीं, ये वाला नहीं, ये वाला नहीं. So that was the lab we have created with this project. So now we will start the scientific session. And the chairperson for the first scientific session is Professor Dr. Tanveer Navid, sir. And our first speaker is Dr. Mallika Chatterjee, Assistant Professor, Amity Institute of Neurophysiology and Neurosciences. So, sir, if you can please take over.
Very good morning to one and all. At the outset, I would like to congratulate Dr. Sudipta Saha and entire team of AIPS for organizing this workshop. We have just witnessed a very well-equipped lab which Dr. Sudipta has created from the fund of this SERB, and this workshop is very much part of this project only. It's my pleasure to introduce the first speaker of the day. And I must say that the first speaker is a young achiever. She is Dr. Mallika Chatterjee. Dr. Mallika currently working in the capacity of assistant professor at MIT Institute of Neuropsychology and Neurosciences, that is the AINN. She obtained her PhD degree from a very reputed institute, University of Connecticut Health Center at USA, and she followed with the postdoctoral research as a very prestigious, Wellcome Trust DBT earlier career fellow at another very reputed institute, that is the Tata Institute of Fundamental Research. Her area of specialization is developmental neurobiology, genetics, system biology, neurodevelopmental disorders. She has published her research in several reputed journals, which include Nature Communication, BMC Biology, Journal of Neuroscience, Developmental Biology, Frontiers in Neurosciences. She is recipient of several prestigious awards. To name a few, she is a recipient of Wellcome Trust DBT India Alliance Early Career Fellowship in 2013, Asia Pacific Development Biology Network Grant for Collaborative Research at Kyoto University, Tokyo, Dr. G.M. Tauri Young Scientist Award from the Society of Neurochemistry, and also International Brain Research Organization, Ghana Foundation Brain Awareness Week Grant. With this, I request Dr. Mallika Chatterjee for her deliberation. Thank you.
Hello, everyone, and thank you, Dr. Saha and IPAS for inviting me to give the first talk. Well, I'm a bit nervous, but to make it a bit easy, I will be asking students questions, and so that, we learn in the process and make it a bit interactive. Okay, so please respond when I ask you questions. So microscopy, as Professor Das correctly pointed out, there's a huge scope in microscopy, and the word lies there, microscope, so scope. And what can we not do with it? As you see here is a picture from my own publication, and it is a mice which has green cells. GFP, again, a word, green fluorescent protein, which Dr. Das pointed out. And it's a very important region of the brain called the thalamus, which acts as a relay station through which sensory information from our various limbs, external like hands, legs, etc. It all gets transmitted to the cortex, to the thalamus. And we in our PhD lab created a mouse line, a transgenic line, which has GFP in its genes. So mice normally do not express GFP, but this particular mouse line has GFP, and that too only in the thalamic cells. I'll show you a little bit more of the pictures soon. But before that, a little bit of theory. I have this question. Do you know what microscopes do? What is the first function that comes across in your mind? Can I have hands up? Anybody? What do you think microscopes do? Anyone? Sorry? Correct. Magnification, right? It makes things bigger. So that is the first thing which comes to our mind. But the question is, does making things bigger the only thing that we need in a microscope? Well, as you see here in the image, this is a little butterfly specimen, and there is this microscope where you have an objective lens, and then you have the condenser lens, and then there's this huge image of a butterfly, right? But in addition to that, in addition to the function of magnification, there is one more function of the microscope, that is resolution. Some of my students are nodding their heads because I think I've done it in their class there. They still remember. But for those who don't know, what is resolution? Resolution is the ability to see 2 points, 2 very closely spaced points, one different from each other. That is, not see them as 1 point, see them as 2 different distinct points. I'll give you an example. If you see a building from afar, say the wall of an Amity building from afar, you will not be able to understand how many bricks are there, right? Because all the bricks are of the same color. But when you come closer to each other, you're going to see many bricks there, right? So your resolution or the capacity to distinguish one brick from another has increased because your distance has decreased towards the object. And that is, mind you, resolution. And in scientific terms, resolution is the minimum distance by which 2 points can be separated and yet still be distinguished as separate points. Now, coming to some formulae, what is resolution? Resolution is 0.61 lambda. What is lambda, anyone? Guess? Wavelength. Okay. Lambda by Na. Now, Na might be a new term. Na is numerical aperture. And obviously, lambda is the wavelength. So resolution simply depends on numerical aperture and the wavelength of the light source as per the formula. Now, what is numerical aperture? In layman's language, it is the light gathering ability of the lens. As you see here, this is a specimen which is lying on the stage of a microscope. You can see the stage here, on this microscope. And I hope all of you have seen a microscope. Any form will always have a stage on which you keep your sample, your slide, etc. Right? Now, above that, so when light is shown, it forms a cone on the specimen. And half angle of that cone is called theta. Mind you, this cone is also called the angular aperture. So Na has a formula again. Numerical aperture is equal to n sine theta. Now, what is small n? Refractive index. Basic 10, 11 physics, everybody knows what is refractive index, right? So now Na depends on what? N and the angular aperture. That is the angle of the light which is shining on the specimen. What is refractive index? Quickly, Snell's law, n1 by n2 is equal to sine theta 2 by sine theta 1, right? And why refraction? Because light bends when it travels from one medium to the other. And what is the medium that we talk about in the microscope? Can anyone say what is the medium in this case? What do you think is the medium? The medium is between the objective lens and the specimen. So what is there in between? Air, right? So yeah, you can use air certainly. Most cases it will work. But air has an N of only 1. N, that is the refractive index, is 1. And If you go with water, that is, you put water between the lens and the specimen, it becomes 1.33. If you put oil, it becomes even more. So what happens to the value of Na if you put water versus oil? Which one will have the higher Na? Oil will have the higher Na, right? So why not use oil? Why do I need to use oil? Now remember, let's go back. Na is equal to n sine theta, and we had R depends on Na inversely. So more the Na value, less the value of R. Now you would say, tell me, R is resolution, and you just told us the greater the resolution, better you will see your images, no? But remember, R is the distance here. That is, even at a very small distance, if you are able to see your objects, that means the object, that lens, particular lens, has very good resolution. So basically, in order to have good resolution, you need to have a smaller value of R, right? Now, if you have a smaller value of R, you will need a larger value of Na. And in order to have a larger value of Na, you need a larger value of N. You just can't change theta that much. You can if it comes very close to the slide, the lens. But other than that, people play around with the value of small n, that is refractive index. So when basically use an oil lens. So that's what people do. When you need to see specimens at a higher resolution, get more clarity, what do? You use the oil lens. That is an N of 1.55 that gives you the maximum resolution possible. So that's a fact. And what is the fact? Greater the value of Na, better is the resolution of a sample. Why am I saying all this? Because you have a very, very precious element of your microscope. Can someone tell me, what do you think? Leave the camera apart, that's an add-on. What is the most precious part of this microscope? Anyone? Lens. It's like a diamond, a girl's most precious, Why is the diamond very expensive? Can anyone say that? It's not rare, right? Why is it expensive? Why is the diamond expensive? Sir, why does DIRF increase? You're correct, but why does it increase? Angle at which the lens is cut. Any glass, or diamond, the angle at which it is cut increases the value of the diamond. Similarly, the lens, in order to shine with a particular angular aperture, needs to be cut at a certain angle. And therefore, the price of the lens is like enormous. All right. So whenever you're working in the lab, be very careful with the lens. They can go up easily from 80,000 and above for a simple microscope as well. This is how this simple lens that you must have seen in a microscope looks like when you open it inside. It has so many different lenses. It is a combination of different types of lenses, which finally gives you this single neat piece of a lens. Okay. And we were talking about refractive index. See here, it's written 0.90. You have, that's the refractive index. 250x is your magnification. All right. So this is an objective lens. 250x is your magnification. To a microscopy expert, the lens is as valuable as a diamond. So be very careful when you work with a microscope lens. Now, there's one more property of light. We all see the simple ray diagram that it meets, at a light meets or is let out from a single point, right? What happens, because there is so much of objects or, things in the air which reflect light, what might happen is light can get obstructed. And that might result in a phenomenon called diffraction. As a result of this diffraction, the light creates something called a ripple. I'll give you an example. If you throw a stone into a water, it not only creates a dent at the point where it touches the water, it also creates waves around it. So that is the ripple. And that is what is diffraction, which each point, when a light shines on an object, each point in the object creates a diffraction. And that was identified by this scientist called George Airy. His surname was Airy, so that's how you get the Airy disk. So light doesn't really end at a point. It creates a diffractive wave or a ripple. And when you go to a more complicated physics, it creates a multi-dimensional 3D, what is called a point spread function. Now, imagine you have a specimen on which there are multiple points, obviously, right? From which light is coming out, and each will create a point spread function. Now, coming back to resolution, you need to see 2 points apart, right? How will you see the 2 points apart? Only when these 2 airy disks do not overlap substantially. That's the only time that you will be able to see resolved 2 points, and therefore your resolution will be best in the resolved case. See here, 2 points, the airy disks are not overlapping. But here they are unresolved. That is 2 points, 2 airy disks overlap, 2 diffraction, those point spread functions overlap, and therefore you are seeing a single point. Okay. So this is you can actually go to, there are various websites which you can go to, like Nikon, Olympus, and Zeiss. These are like the, like the big shots in microscopy. You can go to their websites. They have really good learning tutorials where you can learn about all these, and some of the images are credited to these websites. Okay. Now, this is what I said. Now, in order to account for the diffraction limit, physicist Rayleigh put in a little correction, which was the Rayleigh criterion. We will not go into the details there, but instead of the simple equation that we had, like 0.61 lambda by Na for R, this has now been transformed. You have a new equation to account for the diffraction, that is R equal to 1.22 lambda by Na of both the objective and the condenser lenses. Okay, so that's the new resolution, and anybody can calculate resolution. Today, if you go back to your lab and see a microscope, you'll see the Na written on the lens. You can actually calculate the Ra of the lens, provided you know the wavelength. Okay, now coming back to my favorite subject, neuroscience. So in neuroscience, there are various sized objects, like things as small as, obviously, ion channels, or things as big as relatively the mouse brain. Talking about the R value, the distance at which 2 points can be seen separately, ions and atoms, I'll just give you an idea, has an R value of 0.1 nm. That is at 0.1 nm distance, nanometer distance, they can be seen apart. Whereas in the mouse brain, 100 millimeter distance apart, if these if 2 mouse brains are placed, you can see them. So that is just giving you the extent of resolution. And resolution is certainly needed for distinguishing various nervous system structures. Why? You can decide what lens you have to use in order to see a certain structure. For example, today I give you a 10x lens. Will you be able to see the DNA? What do you think? Just yes, no? No, right? Okay. But with the 10x DNA, will you see a mouse brain? 10x lens, will you see a mouse brain? Should be, right? Yes. So that is how you decide what microscope to use. Now coming back to lambda. We are ignoring lambda right now because Na we are talking about. But R, the resolution depends on lambda. And we all know about the spectrum of light. There is an invisible light, which is the UV, right? 300 to 400 nm. And you have the spectrum from 400 to 700. And then you have infrared or invisible. 5 minutes? Okay, quickly. I'll just maybe skip this part to go to the interesting part of fluorescence. Okay, since we spoke about this, wavelength, that is 300 nm to 800 nm. What is fluorescence? Fluorescence is an emission which is happening because of, sorry, just one second. Yeah, just focus on this diagram. These yellow balls that you see here are basically electrons. A fluorescent molecule absorbs photons from the UV light. UV light is shining, they absorb photons and they emit. They get excited and then they can't remain excited for long. Like us, we have to calm down, right? So the electrons calm down. How? By releasing energy, a photon of lower energy but higher wavelength. So fluorescence emission takes place. That is fluorescence emission, and it is of a very short duration. As seen here, it's only about 10 to the power minus 9 to 10 to the power minus 7 second duration. So it's a very fast emission. Here, UV light, excited filter, it's on a fluorescent specimen like the green brain I showed you, shining on that UV light, and then emitted is blue light. See, there's a shift because UV light is on the left side, which is 300 nm. And when you go to the blue, it comes in the range of 400 to 500. That is longer wavelength. Longer wavelength means low energy. So the fluorescence emission will be of low energy. Quickly into the how fluorescence microscopy came about. This guy, G. Stokes, he described Stokes shift. This shift in energy is called Stokes shift. He described fluorescence. Then fluorescence microscopy came up. Fluorescent dyes binding to living cells first came up. Then finally, this was all happening in vitro. People identified fluorescent molecules in vitro, like chemicals, you can synthesize them. Those were called fluorochromes and fluorophores. Then people identified, for which they got the Nobel Prize in 2008, GFP. And what is landmark? What is the breakthrough? Is that because this was could be used as an in vivo system. Why? Because the GFP gene could be identified and isolated from the jellyfish. And this could be put into other organisms like mice, zebrafish, etc. And they are expressed as a protein and they have their own fluorophore. Therefore, any light falls on these cells, they will fluoresce. Okay. These are the examples of fluorochrome and fluorophore. And since 1962, so many colors of GFP have evolved by mutating 1 or 2 residues in the GFP protein. And you might ask me, what is E? E is the enhanced fluorescence. The fluorescence capability has been enhanced by increasing the half-life, because remember, these are proteins. So you have to stop their degradation. So you increase the half-life by introducing one mutation, and subsequently many such new colors have come up. Okay. So before finishing, I'll just show you 2 more slides. This is an example of our data in the lab, where we actually made that mouse which has only GFP expressing cells in the thalamus region. And this shows a real-time image of the cells of the thalamic brain tissue where nothing else is stained, only the GFP fluorescence is visible. And we have used no immunofluorescence on this. Finally, the crowning glory of neuroscience, where Brainbow mice was discovered. What was Brainbow? By making a transgenic mice in which multiple, by permutation and combination, thousands of colors of various proteins could be made and in the mice itself. So you just took a slice from these mice brains and observe it under the microscope. This is the hippocampal region. And you can see there are so many mice, so many cells which are colored, right? And these are all courtesy the discovery of fluorescence. So first I'll given the, maybe I should stop it, right?
Yeah, in 1 minute maybe. Yeah, in 1 minute. Yes, sure.
Okay. All right. So before going, we have from confocal. So this was fluorescence. Now we do fluorescence with epifluorescence, confocal, and 2-photon microscopes. So the latest in technology, I will. Why do we use confocal? Why don't we not use epifluorescence? Because confocal manages to restrict your light in one plane and therefore give you a much better picture. The picture on the right looks much better than the picture on the left, right? So the left is an epifluorescence picture, the right is a confocal picture, and the resolution or clarity has been immensely increased because of limiting or restricting the light to one plane. Come confocal, after that, is there anything else? Of course, there is one more which I'll end with, which is a 2-photon microscope. Prohibitively expensive, but at the same time gives you beautiful data, because again, it actually is a better bargain than the confocal microscope. Why? Because it restricts the light even more. How? Because it, instead of one light ray shining through, it has 2 low energy light rays shining at the same time on the specimen. And the possibility of 2 low energy light rays shining at the same time on a specimen is very low, the probability, right? Then a one single light ray. So that causes minimal photo bleaching or quenching, as Professor Das was pointing out, and that enormously increases the possibility of live imaging while live samples, because they tend to photo bleach a lot. So these are the advantages of a 2-photon. And because excitation is rare and restricted to a focal point, it causes less photo bleaching and phototoxicity, and particularly useful for long-term imaging of light samples. I will not go into further detail because of lack of time, but you're most welcome to come and ask me questions anytime. And you can come to AINN and ask me questions also, or email me. Okay, thank you.
Hello, is it audible? Yes. Thank you, Dr. Malika, for such wonderful lecture. And in a very simple, interactive way, you have covered the topic. And of course, it was, the presentation was very nice. I don't remember I have seen such colorful and illuminating, presentation. And very nicely you have covered the neuroscience aspect also, because the one slide you have seen, that is of the hippocampus, that was a wonderful. So compliment from all of us for a wonderful presentation. Thank you very much.
Thank you, Dr. Mallika, and thank you, sir, for chairing the session. So our next session is replacement for the hands-on on the immunofluorescence microscope. So the chairperson for the session is Professor Dr. Jaswanta Sethi, and Mr. Bharat Sheth from De Winter's microscope will be coming to give the talk, and it's mostly demonstration.
Since beginning of the talk of our Dean Sahab, Dr. Das, the immunofluorescence microscopy as a very nice topic, and good work done by our Dr. Sudipta Saha in his SERB DST project, followed by a very good presentation I could see and enjoyed, and of course many things to be learned. So now I would like to introduce towards a company manager who is going to present through the microscope a lot for the betterment of all of us. He is none other than Mr. Bharat Sheth, the managing director of De Winter's Optics Private Limited. And this De Winter Optics India is a leading manufacturer, importer, export and service provider of the digital microscope, camera, digital microscopes, fluorescent microscopes, all type of microscopes in industrial as well as academic applications. Of course, they have software is widely used in pharmaceutical, biotechnological, metallurgical, and mineralogical things. Will be definitely will be talked by him, and we are very sure that this is widely used microscopes and to be reproduced as microscopy. He is going to demonstrate on the working and handling of immunofluorescence microscopes. I would like and request Mr. Bharat Sheth to come over the dais and just give us the very good alignment on your work of this. Thank you. Thank you, Mr. Bharat.
Good morning, everyone present over here. I, Vidisha, in behalf of De Winter team, we are glad to get this opportunity of demonstrating our fluorescent microscope in front of you all. So my team would be demonstrating that. So before that, I would like to, before proceeding with the demonstration, I would like to talk about DeWinter, which was established in the year 1989 by Rajiv Luthra, who is the CEO. He is a biotechnology graduate himself. So yeah, so having a 3-decade timeline, corporate timeline, we are proud manufacturers and exporters of a complete range of microscope cameras and software. So now Mr. Bharat Seth and Umesh Kumar would be demonstrating the software, this fluorescent microscope along with the BioWizard software and cameras for better understanding. So, sir, I request you to take over the mic. Well, before that, I would like to present the slide we have prepared. Hello. So yeah, as you have seen the like the presentation we had made regarding the microscope, camera and image analyzing software. So here are our microscopes along with the camera. And this is the fluorescent microscope. So anybody interested can come and operate after study is done with the demonstration. Okay.
This one fluorescence microscope, this one blue filter, and this one TV slide map.
Hello, this is the fluorescence microscope. We attach 5 megapixel camera on that. And this is the live picture coming on. First, TV slide, no problem. You can capture the picture or you can set the camera setting first if you want to change the color setting option. There you can change the color or increase the brightness, contrast, whatever you want to change. You can adjust from here. Okay. Then you want to capture it. Clear the picture. Okay. One push, chalo. Okay. Now we can adjust the color adjustment from here and we transfer the picture.
Dear students, if any
Camera,
question, you can come forward and ask directly.
Normal slide लगा दे, कोई बात नहीं. Normal slide.
Obviously, this is a time-taking procedure. You know, when you are focusing a fluorescent microscope, it takes a lot of time. And once you can focus it, then you can, search the field for a particular like cell or so, and then you can take a picture. It's not that you take a picture like you take in your mobile phone. It is not so easy.
Okay, it's okay. This is the simple slide. We can focus now. And now we can adjust from microscope and capture it. Suppose we can want to capture in measurement, then save it. It will be directly go to the measurement option. Short one कहाँ से होगा? This is the picture. Now you want to measure some dimension like length, angle, curve length like this. First you change the line thickness from here or adjust the font size and change the color. If you want to change that color, okay it. Then length option is there. You want to measure the dia of that particle. This is the length, or you want to curve length is there like this, or results on image. This is the result they are showing, and you can simply click on the report option. Report will be converted into Excel formats. Now I will show you some more slides. This is the suppose count. If you want to count some particle, you can open the picture, adjust the threshold option, compute it, and then they will show you the result. If you want to in area percentage, click on this option. They will distribute it into microns. Suppose 1.5 micron to 357 micron is green color that is shown here. And the bigger one is 1068 micron to 1424 micron, and directly go to report for reporting purpose. Then there is a setting option. You can change from camera, image, set gallery part according to your requirements. Or report will be come in Excel, PDF, HTML in 3 options. That's all. Fluorescence नहीं होगा. Fluorescence slide ही लिखा है. Camera setting here. Focus है तेरा. Okay कर दे. और इस पर वहाँ से. आगे उसमें आगे अब देख करिए ये दिखो आया डिफॉल्ट करूंगा तो नहीं आएगा कलर चेंज कर देता हूँ ओके हाँ बस ठीक है now this is fluorescence light you can see some particles here we can adjust from microscope okay then capture it picture will be captured then go on measurement this is your capture picture and you want to analyze some linear measurement you can draw here some lines like this again you want to take line and results will coming out according to that okay thank you
Thank you, Mr. Bharat and your team members. The demonstration of this immunofluorescence microscope. I believe students enjoyed, and if they have some doubts, you can ask them, ask him, and of course the team members. Thank you, Mr. Bharat.
So if you have any questions, and if you don't have, then we will proceed to the next session. In the next session, that is session number 3, the chairperson is Professor Dr. Subhrajit Biswas, the Deputy Director, Amity Institute of Molecular Medicine and Stem Cell Research, and also the Faculty Coordinator for IPAS. And our speaker will be Dr. Alak Manna from Mayo Clinic, USA. Sir, session is yours.
Thank you, Dr. Sudipto, for introducing me. And I hope Dr. Manna is here.
Dr. Manna is online.
Yes, I am here.
Okay, great. Nice to see you. Yeah, just it's my pleasure to introduce you, Dr. Alak Manna. He is currently working as a research associate scientist in Department of Cancer at Mayo Clinic, Florida, USA. He worked as a postdoctoral fellow at Department of Cancer Biology, Hematology, Oncology, and Mayo Clinic, Jacksonville, Florida. And he worked as a research scientist at the Department of Immunology, Motive Cancer Center, Tampa, USA. And he received his PhD and from University of Calcutta, Department of Biotechnology. So what is his major scientific contribution, which includes the identification of the tumor-associated immunological biomarker for therapeutic intervention, then evaluation of immunotherapy to enhance anti-tumor T-cell immunity in the chronic lymphocytic leukemia, worked on the developing the novel strategies for adopting engineered T-cell based therapy for solid tumors. So today his topic is the application of immunofluorescence in the development of the immunotherapy. So Dr. Manna, yeah, nice to with us. And I'm also introducing myself. I'm Professor Biswas, and I was also in the USA for a decade almost. And in the similar kind of the work like hematology, oncology, especially I was in 2009 to 2012, I was in Vanderbilt Medical Center in hematology and oncology. So maybe even I have visited the Tampa as well as Jacksonville for it. So it's your stage and please go ahead. It's a good initiative by Dr. Sudipto because it's a DST SERB. They have given one the Imbril project and some social responsibility work. So it's called scientific social responsibility. So that is yours and please go ahead. Thank you very much.
Thank you, Dr. Vishwas, for introducing me. So it is a good morning for everyone, and though it's mine is midnight. Okay, so can I share my screen?
Yeah, sure.
Okay, can you see my screen?
Yes, it's visible.
Okay, good. Okay. Okay. A very good morning, everyone. So, and thank you, Dr. Biswas, for some nice introductions for me. And I would like to introduce myself a little bit about my expertise here I'm working with. So as Dr. Biswas said that I'm working with development of immunotherapy. So I will touch everything a little bit. So what I'm doing, but by keeping in mind that the applications of immunopresence, which is theme of this workshop. And congratulations to Dr. Saha for these kinds of initiatives. And I'm going to my for the time constants, I'm just going to my presentations. So as Dr. Chatterjee in the beginning, she explained everything very clearly about the fluorescence, basic principle of fluorescence and how it works. So I'm just touching base about the basic principle of fluorescence, and the main diagram that we remember always in the principle of fluorescence is a Jablonski diagram. That is we learned the biophysics class in the graduation's time, and it's that basic principle of fluorescence is basically the working of the vibrational relaxations of the molecule which is excited by a photon. And when those like the molecule, they are excited to S0 to S1, and they reach to different layer of the vibrational energy. We call it excitation energy. So when that actually basically unstable, and they are coming back to the stable excitation state, and from there they go directly to the S0, which is a ground state, and that actually called fluorescence. If it comes through intersystem crossing, which is called a transition state, so that actually have a phosphorescence. So fluorescence and phosphorescence are basically kind of a similar kind of emission spectrum, but so the principle is completely different. So this is the basic principle of the fluorescence, and then I'm coming to the topics to be presented today in my presentations, that uses of the fluorescence, the biology, which is basics, immunofluorescence, why it is a little bit spatial naming of the fluorescence, and then uses of basic immunofluorescence in biology, applications of immunofluorescence. So I'll mainly concentrate on the applications because that's my work that I'm doing here. So now I'm going to the next one, is the uses of the fluorescence basic biology that everybody was discussing from the morning. I was listening actually. So the tagging of different proteins, biomolecules, lipids with the fluorescence is nowadays like the research tool. And you can see the cellular localizations of biomolecules, cellular integrity by, like I'm giving an example, the GFP, endocytosis, exocytosis, membrane fluidity, enzymatic activity, signal transduction, anything, everything nowadays is basically a fluorescence, like we are using fluorescence in the biology. And also there are different components now widely used, which is a fluorescence microscope, confocal microscope, and also we are using nowadays a fluorescence-based PCR, DNA microarray, DNA sequencing, flow cytometer, fluorescence activated cell sorter, as well as analyzer. And nowadays, recently, we started using a metal-based, instead of fluorescence, we are using metal-based cytometry is called CyTOF. So those are the different type of uses that we are like directly or indirectly using in the biology. So now I'm coming to the immunofluorescence. So what is immunofluorescence? Immunofluorescence, as Dr. B.C. Das was mentioning, that in our childhood that everybody sees about the the fireflies and everything, that actually keeping in mind that so those everybody knows, but immunofluorescence is a little bit different than that. This is a method of biology where we actually tag antibody or protein with some fluorescence molecule, and we can use those to visualize anything, everything on the cell, the tissues, by labeling those. And basically nowadays, antibody are widely fluorescence label, and then that we are using for confocal microscopy, fluorescence microscopy, as well as flow cytometer. So immunofluorescence can be used to see the distribution of proteins, glycans, small biologicals, non-biological molecules. And also immunofluorescence used in visualized structure, such as intermediate like filaments and like actin filaments. So those are nowadays is really useful tool for the research. So I just put some examples how we work. So this is basically a tissue where we identified the PDL1 expression and PD1 expression along with the macrophages. So these are like nowadays tool to quantify the tissues, like some protein expression on the tissues, and also we can get a cell like fluorescence level cells that we can see under microscope, as well as the flow cytometer, which is basically so the difference between the microscopy and the flow cytometer is, in the microscope you can see the detailed structure, localizations of the proteins, and you can really see those. So you can identify basically different components of the cells and compartment of the cells. But the flow cytometer is a little bit wide, so you can use those flow cytometer. I'm sorry. So you can use flow cytometer for identifying different different cell population, as well as multiple cell populations in single tubes. So now I'm coming to the use, examples that we already discussed from the morning, that the detection of a detailed cellular component by fluorescence microscopy. The principle is pretty simple, that you have a light source for exciting the object, and then there will be the emission that will be detected by the detector. It's pretty simple, but and there are the images. So it's basically we're getting something like a 2D pictures, and so but confocal microscopy is a little bit different than the fluorescence microscopy, which is a little bit advanced. So here actually we can play with the planes. So it's basically we fix this plane. If you see the thing, so you can image actually like different layers, and nowadays with the software you can visualize those. So I can share with you one of my example that I did. I took the B cell because I'm working on the B malignant cells. So I took the B cells and tried to understand CD38 and IgM. Is basically I label IgM, and then you can see how IgM level clusters in the cells. I'm just playing the video if you can see it. Yeah, it's basically G-stacking because it took the images and they combine, like they stitching each other and make a video of the different layer. It looks like a 3D picture. So those are the very real thing that you can visualize in terms of the localization of the protein, cluster of different proteins, antigens. So those are the things that we are using nowadays widely, and those are nowadays a tool for the research. I'm sorry, I'm going. So now I'm coming to the flow cytometer. The principle is kind of similar, but here actually we use different beam splitter to split the different beams to the different detector, and then we can use a multiple color in the single populations, and we can actually get them separately to see the different cell populations in single tube. So so this is and the advanced use of the flow cytometer fluorescence in a quantitative way. So you can quantitate the expression of different populations and different proteins on the cells, and how many percentage of the cells are producing some particular protein. So you can differentiate those by flow cytometer, as well as by using this technology, you can separate those particular populations. Suppose if you're working with some populations, because I was working, so the example that I gave here, actually I was working with CD8 positive TSCM with stem cell that we were trying to capture. And you can see the TCM, like central memory cells or stem-like memory cells, we identified by flow cytometer, and we can sort them. So though the population is very low amount in the cells, in the blood, so but still we can understand how many cells are there, and we can sort those cells for further characterizations. Now I'm coming to the immunotherapy. So if you know the history of the immunotherapy, it is not like today's treatment that we are giving. So it is basically Greek physicians Galen started in before 70,000 era, and then it evolves. But in 19th era, it is actually evolved in a way that actually nowadays it's become a very essential tool for the treatment. So the breakthrough was the discovery of interference, which is used for the cancer vaccines by Root and Graham in 1945. Then in 1990, like Dr. E. Donald Thomas got Nobel Prize for bone marrow transplantation. So those are the antibody therapy to the cell-based therapy now evolved the immunotherapy. Now we are in the era of the adoptive cell therapy, which is basically a CAR T. Now the first CAR T was approved in 2018. So now we are there, like with the checkpoint inhibitors and the CAR T. So those are the era we are living now for the treatment. So there is a history of the vaccines. So there are several vaccines which started in 1885 by Dr. Louis Pasteur with the rabies vaccines. Now we are in the era of COVID that everybody know where immunity is and immunotherapy is, what the vaccine is. So that actually evolves. So if we differentiate the types of immunotherapy, there are several types of immunotherapy in practice. This is giving an example of cancer vaccines. There are different cancer vaccines which we are giving in the clinic. It's like Cervarix, Gardasil. Those are actually names for hepatitis B vaccines. And also there are several vaccines for the cancer, which is Provenge for the prostate cancer, T-VAC for the melanoma, and BCG for early stage bladder cancer. So there are different checkpoint inhibitors, as nowadays everybody knows that because in 2018, checkpoint inhibitors got the Nobel Prize. And nowadays we started giving adaptive cell therapy, which is started in 1983. And then in 2018, the first CAR T was approved, which is CD19 CAR T for AML and CML. And then there are different monoclonal antibody treatment, which is widely used for the different cancer, like PD-1 inhibitors, like pembrolizumab, nivolumab. Those are the things they are using as a naked antibody and bispecific antibody, which is called BITE. And also there are oncolytic virus therapy they're using, and immune modulator, which is also a kind of second line of molecule after chemotherapy. And all those research and the treatment are actually like directly, indirectly involves the fluorescence biology and fluorescence principle. So I'm coming to the first applications of immunofluorescence that we use for the development of immunotherapy. The first one is the therapeutic vaccinations that we develop. It's now under the development. So the basic principle we started with is like we collect the blood cells from patients with CLL or multiple myeloma and collect the tumor cells, and then make a apoptotic body by like we make the apoptotic body with, we develop a protocol which actually under patent, so that's why I'm not disclosing how we made the apoptotic body, but we made the apoptotic body, and it's like membrane blades, and then we isolate those membranes, so the apoptotic bodies from here, and then we collect those. Then we characterize those with a flow cytometer. Excuse me. And then we use those, actually we characterize those ABs, and then we prepare for those for the vaccinations. So this is basically a flow chart of the therapeutic vaccinations that we develop. And we first actually identify the different size of the molecule by microscopy, and then we use flow cytometer to characterize those based on their granularity, annexin 5 positivity, and as well as dead cell marker. So you can see those percentage of the cells that we are looking for is 90%, and which is annexin 5 and tau protein. Those are two apoptotic cell marker that we develop as an apoptotic body. Then we use the confocal imaging to characterize those different size of the molecule present in the cells and how their integrity is there, because we label with the different probes like Hex for nucleus and annexin 5 PITC. You can see the first panel, and there are different mode of the production. And then we selected, we label with cytox RNA, which actually label the RNA and annexin 5 with the apoptotic cells. Then we also use a mitotracker gene. The aim was to identify in the apoptotic body that we're generating that how much RNAs are there, how much DNAs are there, how much mitochondria are there, because those things needs to be characterized, because we are giving back to the patients. Though we are giving the autologous setting, so there will be not that much red flag, but still those characterization is needed, because at least FDA needs to know what are those we are giving back to the patient. Then we did like with those apoptotic bodies that we made, we use those for to see the T cell activations, because that actually we are looking for, because those vaccine actually activate the T cells that we anticipated. And we in vitro settings we found that those ABs, which actually therapeutic vaccines that we're developing, so those actually activate T cells. So T cells, it actually induce proliferation of the T cells, and these are the these are the examples that we did by flow, and this is the cumulative data that we got percent proliferations. So DABs and BABs, those are basically two kinds of ABs that we are moving forward with those for the treatment. So those are basically like the proliferation is 40 to 50%. Then we also measure like a thumb rule assay, which is called degranulation assay, which is when we co-culture those ABs with the T cells, and we found when we co-culture with those ABs pulse TC with the target tumor cells, we found those T cells also have CD107 expression on them, which is basically a marker for the T cell activation. So CD107 is basically a LAMP1, which is an indicator for the cytolytic activity of the cells. And when the T cells are expressing CD107, that actually indicates that T cells are activated and cytolytically activated. We did for blood cancer, also as well as solid tumors, and the data was similar. So that's why it proves that even it's better in the solid tumor, if you see here. So the overall T cell activation in when we co-cultured the tumor cells, it was better. So that indicates that those T cells which we activate by the vaccines will be effective against those particular tumors. Then we did animal study where we found that this is the PBS control group. If you see those actually died on day 35. And so this is the last image that we got. And then, we keep a whole tumor cell lysate, like WCL, basically a whole cell lysate. So then we found those also have little background though, but if you compare with AB treated groups, so there is actually it's like a significant difference of the decrease of the tumors. If you see the survival, it's also significant different and also statistically different between these groups, PBS versus WCL, PBS AB versus PBS, AB versus WCL. So all are actually statistically significant. That's actually very, very impressive for us to move forward. Then I'm coming to the second topic that we are working with, the antibody-based immunotherapy. So this is basically one of the cartoons that I made for my publications that we published in 2020 in Blood Advance. So here actually we develop, we work with some antibody which targets CD38. And CD38 is basically approved for multiple myeloma, but nowadays we are trying to use those for other indications like CLL, AML, and pediatric leukemia. So the mechanism that we established by this paper, that those antibody, when we treat in vitro and in vivo, we found the CD8 positive T cells getting activated and it kills the CLL cell. So that's very impressive data that we got with this one. We actually subsequently published three papers with this drug. So, and the drug is known actually as daratumumab, and we found that daratumumab actually, when you treated those cells, and it activates the CD8 positive T cells, and we measured CD69, GITR, CD137, and we found that like daratumumab actually increases. If you see the red dots versus this black dots, so daratumumab is significant CD137 expression, but there is no change of other activation. So it's basically we found that CD137 mediated activations induced by daratumumabs. Then we see the translocation of BEAM and SERB. BEAM is basically an apoptotic protein, and it is after treatment with daratumumab, you see that we label a BEAM with green, red with microfracture, blue is DAPI nucleus 10. If you see that yellow, which is basically red and green, so it's indicated that BEAM is translocated to the mitochondria. That actually indicates that it induces apoptosis. That's the basic mechanism that is known for the B-cell malignancy. That the BCL2 biology, like BCL2 is an anti-apoptotic protein, BEAM and BEAM is basically a pro-apoptotic protein. It's basically the balance game between BCL2 and BEAM, and we found that BEAM translocation increase that indicates that that induces apoptosis. So K is basically curominin, so another small molecule inhibitor for CD38 that is not so significant versus DARA. So that's why it's very, very indicative. And in the second panel that you see that we measure the SERB3. So you see the SERB3 is level with the green. So we found that it actually localized not only in the mitochondria, but also in the nucleus. So activin is basically a deacetylase, which actually deacetylates different proteins to be activated. So when it is localized to the mitochondria, it actually activates several mitochondrial proteins, including BRIM. So that actually helps to be mitochondria mediated apoptosis to be increased. Then we also prove that the daratumumab induced beam translocation and SERB actually leading to the apoptosis. So I just presented a few data here. So if you would like to see a full set of data, you can go and check. It's actually like full PDF available, so you can see, or you can email me. I can share those things that you can see whole set of data here. So here you can see that when we treated them with the daratumumab, there was a significant increase of the apoptosis. So if you see these both panels, it's total about 70%. Then we tested in the mouse model, and you see that data-treated groups, the significant decrease of the tumor versus vehicle. So that actually indicative. So So this is the basically proof. So the image you see here is basically a luminescence that Dr. Das was talking about the luciferase. So we actually use this one here to image the mouse tumor. So it's basically we injected luciferase containing cells, and we give oxyluciferin, d-luciferin. So that actually helps. So that actually helps to image those mouse in vivo. So those are basically example of the firefly oxyluciferin or d-luciferin, which Dr. Das was mentioning in the morning. Then I'm coming to the third section, which I'm currently working with, and we do have 2 clinical trials ongoing with this. So the basic principle of the adaptive cell therapy or cellular therapy is pretty simple. Isolate cells from a patient, engineer those with your identified target. So it's called CAR, chimeric antigen receptor, and then ex vivo expand those engineered CAR cells and give it back to the patients. So the CAR can be different type. So CAR is chimeric antigen receptor, or you can also engineer TCR based on your new antigen that you identified, or you can identify the TIL, tumor infiltrated lymphocyte. Those are main basic soldiers, because those are known to the tumor microenvironment. They actually exhausted partner of the fighting soldiers in the microenvironment. So those could be a very, very potential tool if we can engineer those in the right way. So actually, those are the three groups of the treatment people are working with. So if you overview the treatment modalities of the adaptive cell therapy, so those are the 3 main things are has been noted that I listed also there, the TIL, TCR, and CAR. So if you see that the CAR is the latest one that has been started to show the clinical evidence, but that one is actually first approved. So TIL started by Dr. Rosenberg in NCI, and then TCR started. But still, the CAR, it started later, but it's the winner of the game for now, because you can see the different targets that people are identifying and targeting those by making CAR. So then I'm coming to the different generation of the CAR T that is known, just for an idea. So there's the first generation CAR T, which was first identified. They actually, if you see, this one is basically an antibody which binds the target on the tumor cells, or any other cell that you would like to target. So those are basically the engineering that we are doing. And other part was the CD3 zeta, which is the like a co-stimulatory molecule for the T cells, which is actually the TCR component. And then the second generation CAR was developed. They actually add a co-stimulator, which actually helps to activate the T cells. So then third generation CAR T, and actually they use second co-stimulatory domain to activate more. And fourth generation CAR, they actually use some ARM CAR, which actually they're adding some cytokine tail that can produce lots of cytokine that could be like pro-inflammatory cytokines that could be effective against the tumor. Now we are in the generation of the fifth generation CAR. There actually we are with this single chain variable antibody, then we are having like CD3, costimulatory domain, and also R marker. So you can add anything arm, whichever you like to, along with your card. So this is basically the blueprint of the card design that we are doing now. So these are the variable domain, that binding domain, that you have to identify the target and you have to design those things. And rest of the thing is pretty simple. You can pick anything up from there, because that's pretty thing known. And this part, the last part, which is basically armor, that also you have to choose wisely, which could be better to benefit your car, either car or the tumor microenvironment, or anything else that you would like to target. So this is basically a blueprint of the 5th generation card that we are now working with. So if I'm just giving you just flavor of how we are developing that this single chain variable, which is basically CAR. If you see the binding domain here, so that's basically trickiest part that we have to develop. So we are producing a monoclonal antibody by hybridoma technology. That is actually old technology that people are using. So that's the same technology we are using. So we identify antigen, we immunize the mouse, and then we got different cells in B cells and fused with myeloma cells. And then we select them by head selections. And then we screen those hybridomas for antibody of interest that we are working with. And then we identify those antibody producing hybridoma clones, and then we expand those. And then we identify the monoclonal antibody, and we expand those and making a stock and sending for sequencing. And then we test those antibody for specificity, and then we are getting sequence back from the sequencer, and then we are developing those sequence as a CAR to insert into the CAR construct. And then we are subcloning those CAR construct into CAR T cell, like T cells, and those actually will be treated to the patients. So the workflow of the CAR cell productions is the tumor antigen specific screening, CAR gene construction and viral packaging, and then CAR gene delivery to the T cells, and then CAR T cells validation and preclinical assay, and then give it back to the patient. So these are 5 simple steps that we need. So when we are working with CAR T production, so we are doing from the scratch, and those are the things that the steps of the CAR T productions. So this is one of the example that the new one of the new CAR that is in the pipeline, and we actually started IND filing that actually next year. We are hoping that next year we'll get approval for the first clinical trial to launch. And so this is this basically the cartoon that we doing, that we are getting a leukapheresis from a patient, and then we are arming them with a CAR construct. We are expanding those, and we are storing those, and we are giving back to the patient. So after doing this step, it's basically 14 days step. So we identify CD3 TMEM and transduce the lentivirus. Then we are expanding those CAR-T for 14 days. Those are very crucial because every day we have to monitor those cells, because we have to maintain their functions, viability, and everything. And then we are identifying the CAR. So basically, you have to identify. So we are calling it identity and potency. The first panel is basically identity, is nothing but the CD3 expression. So non-CAR and CAR both, because we are keeping non-CAR also as a control for in vitro assays. And then we are doing a potency test, which is basically we are including one marker with the CAR construct, which in this case actually EGFR. So it's truncated EGFR. We are measuring those in the CAR to see that those CAR construct, how many CAR construct is there in the cells. And then we are doing a functional assay. So there are several functional assays that we are doing with those CAR-T that we are developing. So one is a degranulation assay that I already explained in the vaccines section. So it's basically similar. Here we use different cell line model, and we tested the CD103 expression while we co-culture with them, like the CAR-T, co-culture with them the target tumor cells. So you see there are significant increase of CD107 with the treatment of the CAR-T cells when we target. But when we co-culture with non-target tumor cells, it's basically nothing. So that actually is very, very promising and indicative that it will be target-specific treatment. Then we did functional validations of the CAR-T by enzyme assay, which is also basically use of fluorescence or phosphorescence. So here actually we use a granzyme B with it's called MSD ELISA. It is actually it's basically sulfotag molecule. So it's when we have a linker with a primary antibody and we add analyte here, and actually secondary antibody with the sulfotag. When if this complex forms when your analyte is present, you can see the signal. Otherwise, there is no signal. So that's very specific and sensitive. That's why we were using this. And if you see our data, when we use non-CAR versus CAR, the CAR is significantly increase the granzyme B secretion. So that actually corroborate with the increase of CD107 expression. Granzyme B are one of the molecule that T cell are releasing in the microenvironment when they are functionally cytolytically active. So then we did third assay is direct killing. So we just co-culture them because so here also you can see that if we use flow cytometer, though those target cells are labeled with the GFP. So you can image those, so you can image with the microscopy also, but we did flow because we also have a plan to do actually to see view under microscope. But this was the easiest presentations for us to find that how many cells has been killed by the CAR-T. Then we did again in vivo validations, and you see that this is basically a CAR-T group. It's basically when the CAR-T works, it will be clean mouse. And if you see the non-CAR and previous group, the tumor significantly increases with the time. So, and also it's still ongoing though. I just present my current data. It's still under, we didn't publish this one yet. So this still is ongoing, but still, I mean, so far it is statistically significant, the CAR-T group versus previous and non-CAR. So I would like to conclude these sessions by acknowledging my director for this program that we are running here, Dr. Chin, and Dr. Asacharan Khan. So he was actually my mentor for my vaccine project. Dr. Fabio Malavasi from University of Torino Medical School, Italy. He was my mentor also. We have joint publications with him. Dr. Pinola, so he is my mentor when I was submitting for my K99 grant. And Dr. Kinones Hinojosa, you can see him in the Netflix movie. So he's basically our chair neurosurgery, but he is basically a neurosurgeon and he's famous. So you can see a video surgeon's card in Netflix. So he is the one. He actually we share our lab. We have a collaboration with him, and he's basically a good mentor. And Roxana Isdronka, she's the chair of our hematology oncology department, and I shall be happy to have questions.
Thank you, Dr. Manna. It's exciting talk.
Thank you.
Yeah, it's time for questioning. The students, please come ahead for the questions. Any questions? Okay. Okay, it's a lot of science. Just actually, Dr. Manna, there are a lot of PhD student also here, as well as graduate, postgraduate student also here. So just I want to emphasize one thing, that is you talk about the apoptotic body based the vaccination protocol, right?
Yeah.
On the imaging also. So can you brief little bit a few sentences about the principle of that, so student can understand, I guess.
So actually in the tumor, the T cells are become non-functional. So that's the limitations in the treatment. So the vaccines that we try to develop by doing apoptotic body is the tumor antigen actually needs to be recognized by the T cells. But the way the tumor cell presents its antigen to the T cell, it actually bypass the immune patrolling. So, so by developing the apoptotic body, we actually expose some internal antigen, and that internal antigen will be recognized by the T cells, and the T cells will be activated against those antigens. So therefore, it will kill the tumor cells when they see the tumor.
Right, very nice. So another things actually, okay, student has a question. Yes, please. Yeah, okay, yes. Go ahead, please.
Good afternoon, sir. Good afternoon, sir. I have a question regarding the CAR T cell therapy, which we just saw. That the therapy was very interesting, and I had a question that the CAR T therapy, would it also have cell toxicities if I mean the therapy is just advanced, and would it cause any cell toxicity to any degree?
Maybe he's asking about some side effects.
Yes, sir. I mean,
Yeah.
Yeah, okay, yeah, this is a good point. So actually, so we are normally giving it to autologous settings. So from the same individual, we are collecting the T cells, making it CAR and giving back to that patient. So there is no HLA mismatch and nothing, because we are giving his own cells back to the patient. So then the question will come, if when we are modulating by genetic engineering, those cells could be little bit functionally different, that can have some toxicity. So those are the first thing that we are doing before giving back to the patient. So that's called safety study. That's we are doing in the first. So there is a concern of safety study for the toxicity. Yes. So we are considering those, by keeping in mind that those could the change that we made with the CAR construct, that might have some adverse effect on against normal healthy cells. Yes, possibilities are there, but we are considering those. So we are testing those before giving back to the patient. The second point that I would like to emphasize, this concern will be more when you are giving the allogenic settings. So allogenic settings meaning, because if you think about cancer patient, their T cells are functionally not active that much. So if you collect the T cells and if you change those T cells with the CAR, giving back to the patient, so may not be that much functionally effective if you compare with the healthy donor. So we started thinking about giving allogenic T cells by making it CAR, but the problem is allogenic is more dangerous than the question that you asked for. So that will be more toxic against the other individual. So you have to have HLA phenotype, HLA match. Even in that case, so there is a fair chance of developing GVHD, which is one prime concern while we are giving back to the patient, because GVHD is basically graft versus host disease. So the tumor might be cured, but the patient will die with the GVHD. So that is one of the limitations of the CAR therapy that we are still working on to overcome those.
Okay, great. That is graft versus rejection. That is a major problem in the CAR T cell therapy. So thank you, Alok Manna. Just on any more questions? Just I want to say another thing, that is you have shown antibody based in vivo study also, and a lot of pictures on mice. So can you tell all of us, that is, what is the simplest in vitro assay you are doing for antibody based therapy? For
antibody-based therapy, the simplest assay is ADCC, CDC, ADCP. ADCC is basically antibody-dependent cellular toxicity, antibody-dependent cellular cell toxicity, and CDC is complement-dependent cell toxicity, and CDC. So those are the assays that we are doing for testing the antibody. So actually what we are doing is we are co-culturing T cell versus tumor cells with antibody. So antibody will bind the tumor cells, and then the FC portions will be tagged with either T cells or any other cells. So that actually activates those the T cells and kill the tumor. So this is one of the mechanisms that we are doing in vitro settings. So ADCC or CDC or ADCP, if that antibody actually works with macrophage, then we are checking is ADCP, which is basically antibody-dependent phagocytosis. And the third one is CDC, complement-dependent cytotoxicity. That basically we are giving, it's basically when you are treating your cells with any proteins, there is a fair chance of CDC. So that is also one of the methods that we're looking for in vitro settings.
Thank you very much for your great time. And Dr. Manna is now in midnight, almost 3 a.m. in the morning, I guess. Yes. Thanks a lot, Dr. Manna. Yeah.
You're welcome.
Yeah. So then
Yeah, I mean, if any question is there, so I'm reachable, and Dr. Saha is my brother, so you can reach to him. He is, I mean, also you can reach my emails in the mail email. So my mail email is my last name dot first name at mayo.edu. So any questions that I'll be happy to answer.
great. Thank you. Thank you, Maharaj, very much. So I'm hanging over to Sudipta.
Thank you, Dr. Manna. I know it's a lot of pain in the middle of night. I think
It's okay. Yeah, no problem.
it's around 2:30 a.m. in your place. Yeah, it should be something like that. Okay.
2:50.
Yeah, I know. Thank you so much for giving this talk in my request. So this ends the third session. Before starting the fourth session, I have two things to announce. Number one, after the vote of thanks is over, after the fourth session, I'll be giving the link, the feedback link on the Zoom as well as on the WhatsApp group. So whoever has participated online or offline, you can join there and you can click on the link and you can get an automatic certificate of this seminar signed by our Dean, Professor P.C. Das sir. And and now I would like to welcome on stage our fourth session chairperson, Professor Dr. B.N. Malik, Director, Amity Institute of Neuropsychology and Neurosciences. Welcome, sir. Okay. And I would request my faculty colleague, Dr. Shomabruta Banerjee, to please welcome sir with the bouquet. So in this fourth session, chairperson is Dr. Malik, and the speaker is Dr. Rajkumar Bose from Karolinska Institute, Sweden. Sir, the session is yours.
Hello, good afternoon, everybody. It's my pleasure to be here and to introduce the speaker around. Though this is not my field, but I do use microscopy for some time or other, like many other researchers. So here the speaker, I understand, is Dr. Bose, Dr. Rajkumar Bose. He is there in the Karolinska Institute. He's a big name in the research field in Sweden. He did his graduation from Khulna, Bangladesh. And thereafter, he moved on to Sweden to do his post-graduation, like master's. And from there, he moved on to do the PhD from Sweden, the Karolinska Institute. And after his PhD, he did a couple of not couple, in fact, there are 3 or 4 places he did his postdoc, the way I can understand. That is from Central Michigan University, USA, Umeå University, Sweden, then University of Bergen, and then he has come back to Sweden. His research specialization is a pluripotent stem cell. This is one of the very advanced field, as I can understand, how you can modulate the cell. And certainly you need to have a good microscopy in this field. So let us hear Dr. Bose. Thank you. Yes, Dr. Bose, please.
Okay, thank you very much. Thank you, Dr. Malik. And thank you all who listen to me right now. And it's my pleasure to be in this session. First of all, can you see my slide? Hello?
Not yet. It is something, not yet.
Not yet. Okay.
Have you shared?
Yes, I'm going to share.
Yes, it has come. Now put it in the whole screen mode.
Yes.
That cup type. Yes, now it is okay. Go, please.
Thank you very much. Thank you, Professor Mollik. And thank you all over there who are listening to me. It's my pleasure to be in this session. And this is the title of my talk today. So first of all, I would like to mention that I prepared all slides for beginners to this technique, and how to use this technique, how to look at carefully all these critical steps, and how to handle those troubleshootings when you use this immunocytochemistry technique. And then I will show some immunofluorescence data which are generated in a different project at our labs. So what is immunostaining? It is nothing but an antigen and antibody reaction that can be visualized by a specific color, for example, fluorochrome. And here we see that two antibodies, one of them bind cytoplasmic protein, another one binds nuclear protein, and secondary antibody is conjugated with a specific color. Another marker we can see here, it is called DAFI. That is a nuclear staining that identifies specific cell type. So, and it is important to use DAFI all the time, or HOX all the time when you do the immunostaining, because it will tell you how many cells are there in a specific area. So these are the major techniques of or steps of this technique: cell culture, tissue section, fixation, permeabilization, blocking, antibody incubation, mounting, and imaging. Now I will go a little bit detail each of these steps. So you can use the cells or tissue for this ICC or immunocytochemistry. For example, adherent cells, non-adherent cells, or tissue section. One thing you should keep in mind that if you use non-adherent cells, that basically stem cells, when you work with the stem cells, these are non-adherent cells, and so you need to coat the plate before plating these cells. So how can you do that? Let's say you want to use a 24-well plate. So you can take a sterilized cover slip. This is a small piece of glass, and it normally 13 millimeter glass piece can fit in each of these well, and then you coat this cover slip with a specific coating agent. For example, polyornithine, fibronectin, or laminin, or polyl-lysine or D-lysine. There are several coating reagents you can find in the market. Or if you want to use the tissue section, then you need to follow another protocol, how you should section the tissue on a glass slide. When your cells or tissue is ready for immunocytochemistry, so then you can go for fixation step. So fixation, here you can use a different reagent to fix the cells, but formaldehyde is most commonly used fixating reagent, and it is recommended for membrane and novel antibodies, but it is a cross-linking reagent, so it can reduce signal. In that case, you can use methanol or acetone. For example, some epitope is really sensitive to aldehyde. Sometimes people use a mixture of methanol and acetone. And as I said, so if you use a established protocol, then you do not need to think so much. You just follow it. But if you want to establish a protocol, then you have to optimize the protocol using different fixative chemicals. When you know that your cell is fixed, then you go for permeabilization. As I told you, that if you work with or if you fix your cells or tissue with formaldehyde, then you need to use this permeabilization step with either tritone or methanol. And you can use the concentration 0.1 to 0.4% in PBS, and you can incubate for 10 to 15 minutes at room temperature. One thing you should keep in mind that high concentration or longer incubation may lyse the cells. And you can also fix cells and permeabilize with methanol. Let's say you fix with formaldehyde, you can permeabilize also with methanol as well at room temperature. But the methanol permeabilization is recommended for phosphorylated and nuclear antigen. And longer exposure can damage the cell membrane, microtubules, and other organelles. So then you go for blocking steps. Here you can use different solution to block your sample. Normal serum, bovine serum albumin, and milk powder. So if you use normal serum, the percentage will be 5 to 10% in PBST. If you use BSA, it should be 1 to 5%, milk 1%. It should be non-fat milk. And some other thing you should keep in mind that if you use BSA, it should be free of endogenous IgG. So secondary antibody will not bind with them. And if you use serum, the source of the serum should be as a source of secondary antibody. For example, if you use goat serum, goat as a source of secondary antibody, you should use this serum for blocking. And if you use milk powder, you should not use it for phosphorylated protein, for example, ARC phosphorylation, if you want to test it. So these are the things when you you should remember when you perform the blocking step. Next, I will talk about the antibody. Let's say you have done fixation, blocking. Now you want to do you want to use your primary antibody. And several things you should consider here before selecting the primary antibody. Some, for example, reactivity. And how can you check this reactivity? For example, you use the human primary cells and the antibody you are going to use, you should check whether this antibody has already been tested in human cells or not. If it is tested, then you will see the database in either in the company database or if you search the literature, you will find it, the concentration you should use. But if you do not find any information either in the company database or literature, what you can do, whether you can use this antibody or not in human cell line. Yes, you can do it. In that sense, you have to examine before adding this antibody. For example, you check the epitope sequence of a verified species with the unverified species, in this case here, human cell line. And if it matches, then you can use it. And if it matches over 90%, there is high chance that this antibody will work. The next step is that specificity. The antibody specificity is critical when detecting various post-translational modification, subcellular domains, or specific isoform of a given protein. And there is another issue is that antibody type is monoclonal and polyclonal antibody. A monoclonal antibody produced from a single cell, a single B cell clone, and polyclonal, maybe multiple or mixed. Monoclonal antibody detects a single epitope, and it is highly specific compared to polyclonal. And you can use monoclonal antibody. You do not need to use a lot of monoclonal antibody compared to the polyclonal antibody. So you can think that when you buy the antibody, the monoclonal antibody will be perfect if you can get this from a company. So next, I want to talk about the host. It is also important information when you do the immunocytochemistry, especially when you use the secondary antibody. For example, if you use goat primary antibody, let's say goat beta tubulin 3, then you should use other source of secondary antibody, not goat, maybe rabbit, maybe rat, maybe sheep. That would be sheep or goat, sheep or rabbit anti-mice antibody. Otherwise, you will see the high background and non-specific reaction. And then you should, when you decide that yes, you have find the correct primary antibody and secondary antibody, and then you follow the protocol just to whether you will incubate overnight at 4 degrees centigrade or you will incubate 1 hour at room temperature. So after adding the primary antibody, you wash 3 times before adding the secondary antibody. But before going to the secondary antibody, I will give an example how to buy and select a primary antibody. Let's say you want to buy an antibody called anti-beta tubulin 3 from cell signaling. What you can check on this from this company, you should see the host here, the mouse, and it reacts with human, mouse, rat, and other chemicals. So then you are ready to use either human or mouse or rat cells. You do not need to think about other things or checking anything. You know it works with these cell types or species. The another information here, they mentioned the monoclonal antibody. So this is really good that you can have less background or non-specific activity. And on more information here, it shows that unconjugated. So that means you certainly need a secondary antibody to visualize this antigen antibody reaction. And I will talk about this, why it is important in next slide. And here down there, you can see the concentration. When you see that the company knows that this antibody works with different species, and they also mention the concentration you can use. So now I go to this next, here it's called direct detection and indirect detection. As I told you before, the antibody is conjugated, either primary or secondary. When primary antibody is conjugated, then you can skip the secondary antibody step because it is already conjugated with a specific fluorochrome, so you can detect. And it is called direct detection method. But if the primary antibody is unconjugated, then you definitely need a secondary antibody to look at the result. And that we see in the right side, that they have used this secondary antibody which is conjugated to the specific color. So here is kind of comparison of the direct and indirect detection method based on sensitivity, essay time, multiplexing, background, and flexibility. You can look at the literature, how you can differentiate this. When you know that you have added the primary antibody and secondary antibody, now you go for the counter staining with Duffy. As I told you before, it is a very important step to look at. And you can dilute, if you use Duffy, you can dilute 1 in 1000 and incubate for 5 to 10 minutes. It is okay. And wash 2 times after incubation, and then go for mounting. It is also important that because mounting can protect your cells from dehydration. And you can use this cover slip for a while if you mount the cells. This is an example. You can use the vector shield as a mounting media. So we can refresh our mind what I have told so far. The cell preparation, cell fixation, serum blocking, primary antibody incubation, secondary antibody incubation when primary antibody is not conjugated with the color, and then counter staining and mounting and go for imaging. And I will not talk about the imaging here. You have already got knowledge how you can analyze your data using fluorescent microscope. So now I go to the second part of my talk. Here I will talk about my research, what I'm doing here, and I will present some of my work using this immunocytochemistry method. So before going to that, I would like to present the model and chemical exposure. So chemical exposures here, I used synthetic glucocorticoids called dexamethasone and methyl mercury, but I will talk mainly the glucocorticoids here. So I used to use both in vitro and in vivo model, but I have expertise with in vitro model. So this is one of the model I can present first. It's called human neurospheres. We bought these cells, human embryonic neural stem cells from Lonza. But these cells are prepared from human embryo at gestational week 16, 17, and 19. Sometimes we used to get these cells also from our hospital when they prepared these cells from an aborted child. And you see, these cells can be cultured as 2D or 3D. Here we see the 3D culture of these cells. That's why it's called neurospheres. And here the neurospheres is actually going for differentiating phase because it is culture in absence of growth factor. When you work with stem cells, you always think about the growth factor, because if you want to proliferate the cells, you need to use EGF or FGF that actually proliferate cells, help dividing the cells. But if you want to differentiate the cells, then you remove the proliferating step or growth factor, EGF and FGF. Then you can have either spontaneous differentiation or you can induce differentiation by adding different factors that I will tell you in the next slide. And then you fix the cells and stain with different markers. This is another model I want to talk here that is called primary cortical neural stem cells. I used to prepare these cells from rat embryo at embryonic day 14.5 to 15. And then cells are cultured in vitro condition. And these cells are well characterized. And these cells can give rise to different brain cells, oligodendrocyte, neuron, and astrocyte in presence of a specific inducer. For example, T3 for oligodendrocyte, BDNF for neuron, and CNTF for astrocyte. So here I will present these two papers where I used those model I have already talked to you. But before going to show the result, I want to talk a little bit background of this project. You know, the major changes in the prenatal environment may cause alterations during critical developmental periods with adverse functional consequences later in life. The developing nervous system appears to be especially susceptible to potentially neurotoxic injuries. These studies are focused on the long-lasting effects induced by unfavorable Milo on neural stem cells. So what is glucocorticoids? Glucocorticoids is a lyophilic steroid hormone, and it is secreted from adrenal cortex of the adrenal gland. It has diverse applications as pharmacological or physiological. For example, lung development, or it is being used as immunosuppressive drug. The concern is that excess level of glucocorticoids can have adverse effect. So normally, fetus get lower level of glucocorticoids than mother. It is because an enzyme called 11-beta-hydroxysteroid type 2 dehydrogenase type 2 enzyme. It is located in the placenta that converts cortisol and corticosterone to an inactive products. And the one thing we should remember that this enzyme cannot inhibit transferring cortisol from mother to fetus 100%. So you can, a fetus can get still some cortisol. But when fetus can get high level of cortisol, when the placental function is impaired, let's say this enzyme activity is altered, or pregnant mother secrete high level of cortisols due to severe stress, physiological, psychological, or pregnant mother work in the chemical industry that influence actually releasing this hormone in her body. And or mother get exogenous glucocorticoids in case of preterm delivery or congenital adrenal hyperplasia. It's a genetical disorder that mother cannot generate enough cortisol in the body. So in this sense, we fetus can get high level of cortisol from mother. As I told you that the model I have used in this group project. So first I will present the experimental design of human neural progenitor cells. And it shows here the differentiating and proliferating phase of these cells. And cells, let's say, culture for 5 days, and cells are exposed to 1 micromolar synthetic glucocorticoids called dexamethasone from day 1 to day 5 for differentiating studies, and from day 1 to day 3 for proliferating studies. And I have already mentioned what is proliferating condition and differentiating condition. Then I present the next experimental design where we study long-lasting effects of this drug. And here I use the rat embryonic cortical neural stem cells. So these are the preparating steps of the cells, and cells are then passes to passage 1 and exposed to 1 micromolar dex for 48 hours from day 3 to day 5 in presence of growth factor FGF. So cells can divide. Thereafter, cells passes 2 and passes 3, and without further adding the DEX. And why we wanted to do it? Because we want to study whether DEX has long-lasting effects or not, and DEX in this effect are persistent or not. That's why we call actually daughter cells that may have experience what their parents get. So now I can show some result, and here we see that human neural progenitor cells, and that show that decrease Ki67 compared to the control. And Ki67 is a proliferating marker. It detects mostly all phase of cell cycle. And right side is a statistics. Here it's significantly different of the Ki67. And similar result we found actually when we use rat embryonic cortical neural stem cells, and it shows here down there. And nestin is another marker that actually tell you that cells are in a stem cell condition, or cell, that means these are not in the differentiating phase. And then here we can see that when you differentiate the cells and expose the cells, human embryonic neural progenitor cells show that decrease of neuronal differentiation here and increase astrocyte differentiation that is marked with GFAP. Similar result we see right side here when we use embryonic rat neural progenitor cells. So that means that DEX may have a program effect in this when it is when it gets actually when cells get high level of glucocorticoids. I would not go actually the rest of these figures, because here mainly the purpose to show the application of ICC, and I would go directly to the conclusion. DEX had programming effects in both human and rat NPC. DEX exposure decreased neurogenesis and induced premature astrocyte differentiation. And I go the acknowledgement. I would like to acknowledge Karolinska Institute and Swedish Research Council, because it got funding for these studies. And I'd like to thank my PhD advisor. You can see here, and the Sandra Sakatili and Ola have mentioned on their side, and these are my colleagues. Thank you very much again, all who are listening to me. Thank you.
Yeah, thank you, Dr. Bose. It was a nice elucidation right from the basic technique to coming to your research. Yes, any question from any of these audience, please? Yes, here is Dr. Mallika.
Yeah, I had a question about your those figures actually. There you show that initially neural proliferation is decreased when you overexpress dexamethasone, right? Neural proliferation is decreased.
Yeah.
Am I correct?
Yeah. When cells are exposed to DEX, yes.
Yeah,
Yeah.
so the next figure, you show that astrocytic proliferation, differentiation is happening instead. So if the initial pool of nestin positive progeny is decreased, do you think within that restricted pool there is a fate change, or is it that I'm a bit confused between lack of proliferation versus fate change?
Okay. Yes, what I understood, you wanted to know if it is a differentiating condition, how we can see the effect. You know, when you study the phase, when you study the proliferating phase, that means we add all the time the growth factor. So cells will not change the fate, but when you remove this EGF or FGF, cell will change the phase. Cell will go for differentiating phase, and in that case, let's say we want to study the spontaneous differentiation, and we want to study the effects of these DEX in spontaneous differentiation or fate changing. So that we have done here without adding a specific inducer for differentiating phase, for example, BDNF or CNTF. We just wanted to see the spontaneous differentiation. So if you want to study only the proliferating phase, then you keep adding EGF or FGF when you add the glucocorticoids. But if you go for differentiating, then you should remove this EGF or FGF and you go for spontaneous differentiation. And that's how we did. You got? Yes, that's how we did. That means that we wanted to see whether DEX actually itself can change the neurogenesis or glial differentiation or not. If it is changed, whether it is increasing or decreasing, that we have actually studied. So let's say that we found that when we study the fate changing, that it affects actually fate changing. So then neurogenesis is decreased, but astrocyte is increased. That is not good for the brain development. That I want to actually show in this paper.
All right. Just a question. This is like a gain of function where you've increased the glucocorticoid. I'm wondering what if there's a loss? Do you see a reverse defect in the NSCs?
If you lost what glucocorticoids, you mean?
Yes.
Even less glucocorticoids, or? Okay. So, we have used different concentration of the glucocorticoids in the experimental model, like 10 nanomolar to 1 micromolar. Actually, all of them has effect in the cell proliferation. Particularly, it is called dose-dependent effects of the DEX. And you can see in the literature, someone also has published. Yes, even 10 nanomolar can affect the cell proliferation of the neuron stem cells.
Thank you.
Yes.
Yes, please. Here, Dr. Subrajit.
Yeah, this is nice talk, Dr. Bose. I'll not take too much time. Just I'm curious regarding one scientific question, because Dr. Mallika already talked about technical questions. So you have shown that is glucocorticoid actually altered the senescent portion of the neural stem cells. Okay.
Okay.
Senescence altered the senescence, right? So what kind of the feature will be talking about?
Yeah, that part I did not show it here because I know that this is a seminar for students and they want to know about this method. So I did not talk so much about science, but I can answer you. The senescence that I used in this the paper, different senescence marker. First, we check the Ki67 as I show here. That means that DEX exposure actually decrease cell proliferation, And then we used the polyphyletic marker like to P21, P16 expression by qPCR, and we found that the P16 and P21 expression was down-regulated, up-regulated after DEX exposure. So that, and it is known that they are involved in the cell cycle regulation process. And they are also known as senescence marker. And then we go further, and using this specific senescence marker is called HPon gamma, and also BMI1, and also other HMG1 also. All these markers I have tested in this paper.
Marker tested for to see the self-renewal property of these stem cells?
Yeah, we basically checked it in a polyphyletic condition that this the marker are highly expressed or down-regulated in terms of whether to prove that cells going under senescence or not, and by immunocytochemistry and also by qPCR analysis. Yes.
Great. Thank you very much.
Mm-hmm.
Yes, please. Any other question? Nobody is questioning, or from the back? Yes, the students, you should have some questions. No? Okay, do you have a few minutes that I can? Okay, Dr. Bose, I think you have done a nice presentation, but as you are an expert, and going by the questions, what was going on between Dr. Subrajit, Dr. Mallika, would you comment on these cells as a synchronized cell line or cells? Because unless they are synchronized, a lot of answers depend on that, what they were asking. And second thing, we know that these cortisones have a biphasic response, be it to the lower concentration versus higher, and that, do you have any experience? What should be the low level? What should be the high level of? You said 1 micromolar you have used. If we really talk about there, that should be in nanomolar or maybe even picomolar at certain level. And that is how the diffusion at the developmental stage works. So a lot has to do in that. Do you have any experience on that?
From the honestly, I can tell you the clinical side. I'm an experimental biologist, I will say. So in the cellular model, we know that if we increase concentration higher than 1 micromolar, you may see the non-genomic effect. So there will be no genomic effect. As we know that glucocorticoids works by a glucocorticoid receptor. And if we increase the concentration, probably it will be non-genomic effect. That's why we stick up to the 1 micromolar. And if you have a lower concentration, unfortunately, we have seen even 10 nanomolar concentration, there is some effect in the proliferation. But sometimes we see probably very little change, sometimes a little, no sense. But we see senses is even 10 nanomolar. But 1 micromolar is persistent. We always see these changes happen. And the question about the what concentration or level will be suitable for the development, it is, I really cannot answer this question. Maybe a clinician can answer this. So I'm really sorry about that.
Yeah, I understand. It was not a clinical question. It was more of experimental. I can
experiment. Yeah, that I
only say that at least for noradrenaline, the nanomolar or lower has a proliferating effect as compared to micromolar, where if you go more than 10 micromolar, it goes into apoptosis in a primary cell culture. Okay, leave aside. You have been working in this field. Any comment on primary culture, be it from mouse brain versus chicken brain? Why do you prefer one over other?
Sorry, can you please repeat? You said mouse versus chicken brain. If
Yes,
I use
because there will be a lot of ethical issues So would you
Yeah.
prefer chicken brain? Because you are an expert, you have been working for a
Yeah.
number of years. Any
Yeah.
comment on that?
You know, the ethical issue is, it depends on the country law and ethical committee. So I do not know in India, how is your ethical law? But in Sweden, its ethical law is very strict. If we want to dissect any animal, we have to get permission from the ethical committee. This is one point, but as I compare the human neural progenitor cells with the rat embryonic neural progenitor cells, and we found the similar result. So I do not think that it will be different if we use chicken embryonic cells instead of mice or rat or human. Probably the concentration of the drug might the effect. If you want to see the similar effect, maybe you may need to expose for short time with higher or lower concentration. That one should optimize at the lab. That I want to tell you.
I understand. I appreciate your answers. However, just for the interest of all the students who will be the budding scientists in future, I would just like to add here. No, ethical question is ethics all over the world. And I'm talking about NIH guidelines. Then, if you are trying to take something where the proprioceptors have not been developed, that is 11 days of incubation or when the embryo has grown, then you don't need anything because the receptors have not yet developed. So therefore, if you want to do before that, by 9th day, 10th day of after implantation or the growth, be it chick or mouse or embryo, probably you don't need because the receptors have not grown and then you can culture. Anyway, leave aside that. I have the technical question. I have some clarification from you. Probably it will help the students. When you talk about the two questions, one by one. When you talk about washing, I would think the students should have asked, once you wash, why does the primary antibody doesn't go off? A, and second thing, can the immunological studies or these staining be used as a quantitative assay, or they are only qualitative?
Yes, that's why people use this. If you want to do quantitative assay, you can go for this fact sorting, as you know, and you can see that the percentage of the specific antigen antibody binding cells. And if you go for what other question? Sorry.
That's okay. I thought the students will be more interested in that. I appreciate your answers, and thank you very much on behalf of the audience here, on my behalf, as well as the organizers' behalf. I thank you for your presentation, Dr. Bose.
Yeah, thank you. Thank you too.
Thank you, Dr. Bose, and thank you, sir. So here we come to the end of our 4 sessions and this workshop. So before that, I would request my faculty colleague, Dr. Sandhya Singh, to please come up to give the vote of thanks.
Very good afternoon to one and all present here. On the behalf of organizing team, I would like to thank the eminent speaker who took their time out from their busy schedule and spoke to us about their knowledge and wisdom. Moreover, I will also like to thank the chairpersons of every session. They made the talk even more interesting by asking very basic question and very applied question. Also, my heartfelt thanks to the founder president, Ashok K. Chauhan, Vice Chancellor, Dr. Atul Chauhan, and our very active Vice Chancellor, Balwinder Shukla ma'am, for constantly motivating us for striving to excellence.
Also, I will like to thank our Dean, Professor B.C. Das, our faculty coordinator, Suprajit Vishwas sir, not only for spending time with us today, but also helping us, guiding us all the time. I will also like to thank the funding body, DST SERB, who funded this workshop under the theme of scientific social responsibility for the project sanctioned to Saha sir. And I hope to see such workshop more in the futures. Last but not the least, I will also like to thank our faculty colleague, Mrs. Lakshmi Ramesh, Dr. Sudipto Saha, Dr. Soumya, Dr. Harshavij, for helping in coordinating this workshop, making it a successful. Last but not the least, a very big thanks to all the participants for patient listening. And I hope all of you will get some benefit from this knowledge which you have gained today, and to make this workshop event a successful one. Thank you so much, one and all.
Okay, so before we go out for lunch, there are two announcements I need to make. One is that we will have a group photograph on the F2 foyer, and that is one. Another one is in the lunch section, as the information came to me, there is a separate section made for the registered students, and for the speakers, chairpersons and faculties, there is another section made. So kindly follow the instructions on the over there. And so let us, so I would announce that the program ends here. So let us go off for the group photograph first, and then we will have our lunch. And I have already sent out the certificate link on the WhatsApp group. I will be just now sending out the certificate link on the Zoom chat box, and this will be active for another 5 minutes. So please click on that, and the certificate will automatically go to your emails. Okay, thank you so much. Thank you for attending the workshop. Thank you so much.